Hello, everyone. Good evening and welcome. I'm Alexander Hoffmann. I'm the director of the Institute for Quantitative and Computational Biosciences and a professor in the Department of Microbiology, Immunology, and Molecular Genetics. Before we begin, I wanted to mention a few housekeeping items. This event is being recorded, and in addition, the chat function has been disabled, but you may submit questions during the webinar using the Q&A function at the bottom of your screen. Today, I welcome you to our webinar hosted by the Institute for Quantitative and Computational Biosciences, also known as QCBio. As many of you appreciate, big data and computation have profoundly affected all aspects of our lives and also all academic disciplines. In the biomedical sciences, this development amounts to what can be really described as a revolution. 20 years ago, computational work in biomedical sciences probably occupied 5% of our research efforts. Today, it is more than 50%. This radical change affects not just how we do research, but how we train our students to do research, and indeed what we teach to our undergraduates. To guide this massive transformation at UCLA, the Institute for Quantitative and Computational Biosciences was established in 2014 and covers research training and undergraduate education. The Institute, QCBio, is a partnership between UCLA Life Sciences, Physical Sciences, the Health Sciences, and Engineering. It involves faculty, postdocs, graduate students, undergraduates from more than 15 departments, ranging from microbiology and immunology, my department, to physics, to computer science, to pathology in a medical school. And what unifies all of these partners is the mission to expand quantitative and computational biosciences research, training, and education. And with the revolution in AI, these changes, how we do research and what we train our students, just continues. In today's event, Age Just a Number, we will hear from 3 esteemed professors from different departments across campus. They will describe and discuss exciting discoveries, such as the existence of an epigenetic aging clock, longevity metabolites, as well as the development of interventions that may have the potential to dramatically extend human health spans and lifespans. Following their presentations, I'll join them to pose your questions. We look forward to taking questions from the audience, which you can submit through the Q&A feature at the bottom of your screen. Please feel free to submit your questions anytime, and note that the chat function is turned off. It is my great pleasure to introduce the 3 faculty who will be speaking with us this evening. First, I'm pleased to introduce you to Matteo Pellegrini. Dr. Matteo Pellegrini is a biophysicist by training who has served on the UCLA Life Sciences Division faculty since he joined the Department of Molecular, Cell, and Developmental Biology in 2005. Dr. Pellegrini earned his bachelor's degree in physics at Columbia University, his PhD in physics at Stanford, and he was a postdoctoral fellow at UCLA, where he worked on computational biology. Following his postdoctoral studies, Dr. Pellegrini co-founded a startup company and later worked for the pharmaceutical company Merck, before returning to UCLA as a professor. His laboratory research centers on the development of a novel computational approach, or multiple approaches, to analyze large-scale genomic data. Please join me in welcoming Dr. Pellegrini.
Okay, so today I'm going to tell you about some of our recent work studying what we call the molecular lifespan of species. So what do we mean by molecular lifespan of species? Well, first of all, we have to realize that the lifespan of a species is sort of the average length of time that the typical member of that species lives, right? but the average lifespan of an individual depends on many factors. And some of those factors are sort of intrinsic, they're genetic factors, molecular factors, and other factors are extrinsic, so the environment and a disease and other factors. So we know that lifespan is greatly influenced by environmental factors, and the simplest manifestation of that is that if we look at the human species, our lifespan has gone from 30 years to over 80 years in the span of a couple centuries. And so just as a sort of an analogy, we can think of the lifespan as a video game, right? Imagine you're playing a video game where you have to run away from monsters, and as soon as you lose the game is when you fall off a bridge or something like that. So as somebody gets better at playing the video game, they can live longer and longer in the video game. And that's sort of an analogy for what's happened with the human species, is we've figured out how to combat disease and live longer. In contrast to this, though, there's also an intrinsic factor that determines the lifespan of species, and that has to do with our genetics. And this genetic factor is extremely important. It creates dramatically different lifespans across mammals. So we know that very small, short-lived mammals live 1 or 2 years, things like weasels and mice and things like that. Whereas other animals live very long. The longest is the bowhead whale that lives over 200 years. And of course, these animals have the same environmental factors that humans have. So some of them may have short lifespan because of predation or because of disease or changing environments, etc. So back to our analogy of a video game, they're playing the same game that we're playing, but potentially they have different hardware. So imagine playing the same video game on a computer that runs slowly. The amount of time it would take you to die in the video game would be longer, right? Because the clock of the computer is running more slowly, whereas on a more modern, faster computer, you would die more quickly. And so the analogy is that independently of the environment, there's some intrinsic thing that's driving this rate of aging across species. Just as, there's an inherent clock that determines how fast the CPU of a computer runs. So how do we identify what this molecular driver of lifespan is? After many years of research, the most promising candidate for a molecular mark that can track the lifespan of a species happens to be directly on our DNA. But our DNA is unchanging. From birth to death, the sequence of our DNA is unchanging. But what's changing is the way the DNA is interacting with other molecules and is covalently modified within our cells. And the specific molecular modification that we focused on is called DNA methylation, which is represented by these little hexagons. So these are small carbon molecules that are attached to our DNA. And so the analogy here is, imagine, our genome is a long document with billions of letters, but it's heavily redacted. So at any one time, cells are only reading part of the genome, just as we would read only part of a redacted document. And the DNA methylation you can think of as the redaction, so silencing certain parts of DNA so they can't be read. And what we find is these patterns of silencing are changing throughout our lifespan. And so what this leads to is the creation of what's been referred to as an epigenetic clock or an epigenetic trajectory. So in this graph on the left, we see the age of individuals. These are humans from 0 to 100 years on the x-axis. And on the y-axis, we see their epigenetic age. So this is a manifestation of the DNA methylation level across their genome, not at just one site, but at many sites. And we can see that the two things track each other perfectly, so that the epigenetic age is very closely correlated to the actual age. But if you look more carefully at this curve, what you see is that this relationship is not linear. It's non-linear. So what this tells us is that early in life, our DNA methylation is changing rapidly. But as we age, it changes more and more slowly. And it follows a form that in mathematics we call an exponential form. And this is reminiscent to something we're all very familiar with, which is radioactivity, right? We know that radioactivity of any element will decay exponentially as the number of atoms change. And so the for any type of exponential process, like a radioactive decay, there's what we call a half-life or a time scale, which is the time it takes for half the atoms in the case of radioactivity to decay. And similarly, in the case of a lifespan, we have this exponential time scale that defines how quickly the epigenetic age is changing throughout our lifespan. So we looked at this phenomenon across species, and we looked at mice, and we looked at killer whales, and we looked at horses and humans and dozens of other species. And we find that in all of them, we see the same pattern, that the DNA methylation is changing early in life, then it slows down, and it follows this exponential pattern. So this tells us that there's, just like in a computer that may be changing the speed of its CPU, across species, there's some intrinsic process that's driving the same change in DNA methylation at different rates. And we can quantify this with a simple equation that's what we call a saturating exponential equation. But the only point I want to make here is that this is kind of a universal equation that allows us to describe this single chemical reaction across species. And the only thing that changes across these species is their lifespan. And so what we can do is we can use this equation then to estimate what the intrinsic lifespan of a species is. That is, what is their molecular lifespan that doesn't depend on their environment. So it doesn't depend on whether there was more predation, whether there was more disease. It's just sort of the intrinsic rate at which DNA methylation is changing that is associated with their lifespan. And when we look at this equation, we find that humans, in fact, have an intrinsic lifespan of approximately 80 years, which is actually very close to the global lifespan that we've achieved as we've reduced disease burden. And we see that for other species, as we expect, cats seem to live a little bit longer than dogs, and et cetera, et cetera. So there's a broad range of these molecular lifespans, and they seem to track the actual measured lifespans that we know very closely. So the next question is, what drives the changes in these molecular lifespans across species? And for many years, we've known that lifespan is intrinsically related to metabolic rate. So this is how rapidly cells are consuming energy. And in fact, what we know is that large animals have slower metabolic rates and longer lifespans. That is, they need to burn fewer calories because they have smaller surface area to volume, and so they can live much longer with slower metabolism. And so this brings us to the hypothesis that what's probably regulating these chemical reactions associated with lifespan and DNA methylation are fundamentally linked to our metabolism, which is inevitably fundamentally linked to our mitochondria, which are the organelles in the cells that are responsible for regulating metabolism. So the last question is this rate alterable, right? So we know that this rate is different across species, but within a species, can we change this inherent molecular rate that's associated with our lifespan? And the answer is yes, in certain species. So for instance, in mice, we've known for a long time that if you feed the mice fewer calories, they tend to live longer. And when we looked at these molecular clocks in mice, we find that, in fact, it's not just that the mice are living longer with lower calories, but their molecular clock, their epigenetic clock, is actually ticking more slowly. So they're aging more slowly at a molecular level. They're not just living longer. And another example was a study we did of marmots. So marmots are animals that spend about half their life hibernating. And so while they're hibernating, they're essentially sleeping at extremely low temperatures, and their metabolism goes close to zero. It's like living in a freezer practically. And what we find is if we look at the molecular clock, it ticks, it sort of has a yearly oscillation, and during their hibernation time, it practically goes to zero. So what this tells us is that marmots essentially only age when they're awake, and while they're sleeping, while they're hibernating, they're not aging at all. So this again tells us that this molecular reaction, this metabolic reaction, is alterable by environment if we can alter this fundamental metabolic rate. So just to conclude then, hopefully I've convinced you that there is a way to estimate the intrinsic lifespan of a species that's independent of the environment by looking at these molecular reactions. And so from these species, then we can predict what the inherent molecular lifespan of a species is. And for humans, it's about 80 years. And we've shown that these rates of molecular clocks are likely regulated by some kind of metabolic process, probably regulated by the mitochondria, and that we can alter them, at least we've shown in mice and marmots. We can alter this by reducing their caloric intake, or by forcing them to hibernate, or letting them hibernate during the year. So the last question, which I won't answer, but the next two speakers will touch on, is what about humans? Can we slow these molecular clocks in humans, and can we make humans live longer by altering their molecular process rather than just by curing disease and reducing death by disease? I'll stop there.
That was super interesting, Matteo. I think so remarkable that there is this intrinsic clock, and it sounds daunting that, you know, that age and the march of time just goes forward. I wish we could hibernate once in a while. But before we go into the discussion, let me introduce our next speaker, who's Dr. Jing Huang. And I'm really pleased to introduce her. Dr. Jing Huang joined UCLA as a professor of molecular and medical pharmacology in the David Geffen School of Medicine in 2001. So she's been at UCLA for over 20 years. She earned a bachelor degree in genetics, in genetic engineering, actually, at Fudan University, and her PhD in biochemistry and molecular biology at USC, the Keck School of Medicine. And then she did her postdoctoral work in chemistry and chemical biology at Harvard University. Dr. Huang uses functional genomics, proteomics, chemical genetics, molecular biology, biochemistry, and bioinformatics to study the molecular basis of aging and how it is regulated, in the hope of creating therapies for the prevention and treatment of age-related diseases, including cancer, heart disease, and neurodegenerative disease. And so please join me in welcoming Dr. Huang.
So in the lab, we study aging. And as Matteo has already mentioned, these are some of the fundamental questions that we ask in the field. When we first started, actually, other than that, it's interesting for theoretical investigations about the causes of aging and how aging could be regulated. There's also the question of the question of being able to, because aging is a main cause for a lot of the common diseases. So the idea is if we are able to inactivate aging or prevent aging, the idea is we will be able to prevent all these common diseases. So can aging be slowed? The answer is definitely yes. Matteo talked about calorie restriction, and calorie restriction has been shown to be a very robust method to delay aging in almost all the animals. In all the animals tested, it is unknown for human, but in primates, if you restrict their dietary intake, you can see that they age much slower. And but calorie restriction is also very hard to do. I don't know whether any one of you have done that, but it's not very convenient. And also there could be psychological influences because you'll be thinking about, because you're hungry and you'll be thinking about food all the time. But there's also indication that genes can dramatically modify lifespan. And this first publication came from this humble worm C. elegans, where it was discovered that the mutation of a single gene, in this case, BAF2, which is a homolog of human insulin and insulin-like growth factor 1 receptor. If you have mutation in this gene, you will actually live twice as long. And there are other genetic mutations in mitochondrial regulation that could do similar things. If for the dietary restriction, it's very interesting that pharmacologically, you could mimic dietary restriction by targeting, for example, mTOR, in this case with rapamycin, that mTOR is a mediator of the effects of dietary restriction. And if you treat animals with, from yeast to mouse with rapamycin, you can delay aging. And we're very interested in answering the question of whether, because all these are linked. Oh, I forgot to mention that for the DAF2 in human studies in genetics have told us that these same variants in the IGF-1 receptor and the downstream FOXO3 variants are associated with exceptional longevity in the population all over the world. So all these are related to metabolism, and we would like to know, at the time we would like to ask the question of whether there is actually endogenous signals that allow us to do this instead of staying hungry. But if you do stay hungry, if you're able to stay hungry, you will stay young. So we look at the metabolites, and the answer, of course, was yes. This was work by a graduate student in the lab, Rando. Rando found that if you treat the worms with alpha-ketoglutarate, you can dramatically increase their lifespan by about 70%. And the interesting thing is, if you give the worms that had the insulin receptor mutation that live very long already, it can still increase lifespan. And what we're interested in knowing is what is the mechanism of action for these, because if you're able to do this, you will be able to help understand the mechanism of aging, and you will also be able to design better therapeutics for aging and aging-related diseases. And we use this method developed in a lab called the DARTS that allows us to look at how alpha-KG may be able to slow aging. And to make a long story short, we found that a mitochondrial component, mitochondrial complex 5, is a direct target of alpha-KG, and that leads to decreased TOR signaling, and which is a negative regulator of aging. And so it explains some of the findings that we have, that if you treat cells or organisms with alpha-KG, you actually have a slower aging and a lack of tumor formation. And alpha-KG induces autophagy, which is a hallmark of dietary restriction and mTOR inhibition. In this case, we found that through these kind of studies that induction of autophagy by alpha-KG and actually by mTOR and also complex 5 inhibitor oligomycin could reactivate dormant hair follicle stem cells and lead to hair regeneration. In this case, you can see that alpha-KG treated skin also have thicker dermal layer and more collagen. Alpha-KG also has a lot of other beneficial effects in many different systems, which I won't be able, looking at my timer, I won't be able to share today. And alpha-KG is not actually the only metabolite we found that could slow aging. For example, here's another avocado acid called avoketobutyrate, and we found that you can treat really old mice, 90 weeks or close to probably 75 or 80. 80-year-old human, you can treat them with avokb, and you find that they will actually look younger and they also live longer. And this metabolite also acts by regulating metabolism, in this case, actually for AMPK. And here you can see that in the muscle, this is for worms. In the muscle, in the aged animals, you will see this more elongated network structure as in the young worms. When the worms get older, they actually lost this web-like structure and become more punctate and isolated. So there is no question that you can slow aging, and there's also some indications that perhaps aging may be reversible, through the rejuvenating effects of things like dietary restriction and pharmacological intervention. So here are our lab members and collaborators, without whom none of these could be possible. Thank you.
Thanks so much, Jing. That's super interesting. It sounds like a really powerful metabolite. I'm sure we're going to have questions about where can I buy this and is it safe? So we'll talk about that in a minute. Thank you so much for that contribution. Now, let me introduce Dr. Tom Rando. It's my distinct pleasure to do so. Dr. Rando is the director of the Broad Stem Cell Research Center at UCLA. And so it's a particular pleasure to have him participate in our event today. He received his bachelor degree in biochemistry and a doctorate in cell and developmental biology and a medical degree at Harvard University, and then completed his internship at Mass General Hospital. He did his residency in neurology at UCSF and a research fellowship in molecular pharmacology at Stanford University. Research in the Rando lab lies in the intersection of basic stem cell biology and the biology of aging. And Dr. Rando's research has revealed how stem cells respond to cues from their environment to modulate their ability to maintain tissues and engage in tissue repair. His laboratory is credited with pioneering work in stem cell aging and revealed how factors in blood can promote stem cell activity in young individuals and suppress it in older individuals, which I think is what we'll hear about. This work has led to novel clinical trials for age-related disorders, and Dr. Rando has published over 200 peer-reviewed articles and has trained over 100 students, fellows, and visiting scholars, most of whom continue biomedical research. So please join me in welcoming Dr. Rando.
Great. Thank you so much. Again, it's a real pleasure to be here. And I will talk today about the rate of aging also. I'll build upon some of the results that Dr. Pellegrini and Dr. Huang mentioned, and I'll talk a little bit about some of the other work that we're doing. So it's obviously been said that humans are living longer now than ever, and you read this always in the newspapers, and there's much evidence of that. But what does that really mean? I just want to illustrate with this one slide from a book by Leonard Hayflick, who's a renowned gerontologist, on how and why we age. And what he has done in this figure is illustrated how human aging has changed over the millennia. I mean, starting with this dotted line, this speculation that tens of thousands of years ago, humans, like animals in the wild, had a huge amount of early death. death in infancy, childhood, early adulthood. And basically that just declined over time, very much like we see with animals in the wild, this kind of exponential decay. But with the advent of agriculture and civilization, many more individuals were allowed to stay alive, basically were promoted to stay alive during early adulthood, but then again began to die later in life. And this last curve is from 20th century US kind of data, where there's still a little bit of infant mortality, but most people survive through to middle age and then start dying into old age. And as you can see, really this median lifespan, which has, as Dr. Pellegrini mentioned, increased dramatically over the millennia, is in large part, and one of the members of the audience asked this question, due to this incredible increase in survival of young people, infants and children. So the question is really, as we get toward middle age, are we really still aging more slowly at that point? And here's some data from the CDC, which was asking for people across the US from the early 1900s to the early 2000s, if you reach 60, how many more years of life can you expect? In the early 1900s, that was around 15 extra years for males and females. And you can see over the century, that number has just continued to increase. So a person reaching the age of 60 in the early 2000s could be expected to have upwards of 20 to 25 more years of life. So it really does suggest, in a sense, that over the millennia, as we have increased median lifespan, we're also, in a sense, aging more slowly, even into adulthood. And one can see that sort of anecdotally with icons from different centuries. So this is two pictures of Charles Darwin from the 19th century, lived in the mid-1800s. And here's Charles Darwin as an adult, and then here as an older individual. And I think through the lens of the eyes of someone in the 21st century, if you were to predict how old he was in these two pictures, you might be surprised to find out that he was only 45 in this picture and 64 in this picture. And again, anecdotally, if we look at an icon from the 20th century of exactly the same age, these are images of Tina Turner of exactly the same age. And so this is the sort of proverbial 60 is the new 40. And clearly people are seeming to age healthier and longer with time. So the question that I'll ask is, as the previous speakers have addressed, is can the rate of aging be changed? Now, can it be accelerated? We might not want that, but can it be? Can it be slowed? And then I'll end with, can it be reversed? So can aging be accelerated? Well, there are genetic diseases where this is clearly the case. So here's a young woman with a disease called Werner's syndrome. Here she is at 15 years, had perfectly normal development. And here she is some years later. And again, I think you'd be surprised to know that her age at this point was 48. So this is one of many of these so-called progerias, which are these genetic diseases in which people age much more rapidly. And they see accelerated aging really across many, many cells and tissues across the body. So these are genetic diseases, but clearly there are lifestyle aspects that can also change the rate of aging, including accelerating the rate of aging. So if one thinks of poor diet, poor habits, one can, again, anecdotally note that one can clearly accelerate one's rate of aging depending on how one spends one's time. So I refer to this as the Stone syndrome. So, of course, we're not interested in accelerating aging. What we're interested in is, can we slow it down and even arrest it? So this is, again, data that both speakers have referred to in terms of caloric restriction. So this is a paper published by a colleague, Dr. Weindruck, in 1986, in which he looked at the lifespan of mice under different states of diet and dietary restriction. So this green line is the control mice, with about half the mice living to just under 30 months of age, and the oldest mice living to about 35, 36 months of age. But with calories restricted by 25%, which is really quite a bit, already there's a dramatic increase in lifespan where upwards of 15 or 20% more of the animals are alive at the same age. With an even greater restriction of calories, and of course this is extreme, 55 or 65%, they could see really almost a 50% increase in the median lifespan of mice, as well as the maximum lifespan. So this is clearly a very profound way to extend lifespan and indeed health span. And in fact, if you want, you can join the Caloric Restriction Society, and you can restrict your calories, and you can hope that all that hunger over all those years would actually result in a longer and healthier life. But we really have no data on this in humans. As Dr. Huang mentioned, this drug rapamycin was discovered on Easter Island in the soil there. The native name is Rapa Nui, which is where rapamycin comes from. And rapamycin has been shown to extend lifespan in a whole variety of organisms. Here's some data from an important paper in 2009 in which the investigators actually gave rapamycin to mice, not from birth, but actually starting at middle age. And in the blue line, you see the control curve of the lifespan of control mice. But in the mice treated with rapamycin, again, they lived longer, both median lifespan and maximum lifespan. So this is a pharmacologic way to introduce a way to expand lifespan at both median and maximum lifespan. And there are people now who are taking rapamycin off-label on the hope, again, that this will extend their health span and their lifespan. So finally, can aging be reversed? And this is, of course, a very controversial area of research. It's an important area to kind of consider biologically. And my lab got into this in the early 2000s when we were doing these experiments with so-called parabiosis. So these are experiments where mice are surgically connected, and they develop a single shared circulatory system over the course of a few weeks. And so we connected both young mice as controls and old mice as controls. But the interesting pairs are these young mice connected to old mice, which we would leave them connected for several months and then analyze the tissues of either the young or old mice. And what we found to our surprise was that the old mice appear to get younger and the young mice appear to get older. So there appear to be factors coming from the young animal that actually seemingly rejuvenate the tissue of the old mouse. And there are factors from the old mouse that are accelerating the rate of aging of the young mouse. And this is just one experimental result, which I won't go into the details, but we're looking at muscle repair here, either in the young or the old controls. And this whitish area is the kind of fibrosis or scarring that you see in old tissues. You see basically none of that in the young, but in an old animal that has been exposed to the circulating environment of a young partner, you see basically muscle repair that looks just like young animals. So this is really kind of evidence that the tissue appears to have reacquired youthful properties. So we studied this initially in muscle and liver, and then later in brain. And then over the last decade or so, many investigators have used this technique to show that aged tissues, a variety of tissues, can reacquire youthful properties through this technique of parabiosis. And in 2017, we made it to prime time when this idea was picked up by the sitcom Silicon Valley, and one of the lead characters was hiring these young buff males to give him blood transfusions on, again, the hope that this will lead to a longer, healthier life. So is this really rejuvenation? This is the kind of question that we get to in the laboratory. Are we truly reversing the aging process? Can we use these kind of molecular clocks that Dr. Pellegrini referred to understand the process of aging and then restoration of youthfulness? And I think this is still a very active area of research. And there are other ways that people are looking at rejuvenation, including pharmacologic and genetic ways to do that. So with that, I will stop and hand the microphone back to Dr. Hoffmann and look forward to the Q&A.
Oh, thanks so much, Dr. Rando. This is really amazing. I think we have a lot to discuss, and we have just a few, well, about 15 minutes. And so why don't I invite, Dr. Pellegrini and Dr. Huang back here. It strikes me from looking at the graphs, Tom, that you showed at the beginning, at the very first slide, that lifespan used to be primarily limited by, not by aging, but by really health problems, right? And now that many of those have been addressed by medicine, and we have this incredible extension of lifespan, now aging becomes much more prominent. Would you agree with that? Or
yeah, I would certainly agree with that. I mean, in fact, the evolutionary theorists would argue that aging did not exist over evolutionary time because animals lived long enough to reproduce, and they were subject to predation and starvation and dehydration. And so basically across all species, aging wasn't even a feature of animals. And so it is true that with both kind of societal changes like better food preservation, refrigeration, vaccination. I mean, these have been really critically important in getting newborns to adulthood. Now that we can get them into adulthood, now we're seeing the effects of aging, and we're starting to, again, and we see this also in animals in the zoo, in animals in the lab, in animals on the farm. So when there's protection against the wild, then we start to see all these features of aging that we study.
Right, right. And so aside from measuring somebody's health, we also actually need to be able to measure somebody's age, which is where the epigenetic or this epigenetic measurement that Matteo was talking about, maybe is that the best way to measure aging then? Is that what we have accessible? Matteo, is this way of measuring also something that we can address in a per person basis? So when Tom showed pictures of Charles Darwin versus Tina Turner or others, would those people who appear older at the same age, would their epigenetic clock be different?
Well, that's the big question. So, I mean, the industry is searching for what we refer to as the biological age, a way to measure biological age, right? We all know our chronological age, but what we really want to know is our biological age. Like, are we, aging more rapidly or more slowly than the typical person of our age? And obviously, any one measure is probably not going to get you that. You need some kind of comprehensive evaluation with lots of molecular measures. And there certainly is becoming a big industry. There are lots of companies that are selling epigenetic clocks. And of course, there are other molecular markers that are strongly associated with aging, metabolomics, proteomics, all kinds of techniques. So I think in the future, beyond the standard assays that we get when we go to the doctor, right? Blood pressure, cholesterol, these kind of things. It may be that the doctor will run a panel of very sort of sophisticated, high-dimensional measurements that don't just tell you, about single things, but give you an overall evaluation of your rate of aging based on different things. And I don't think we're there yet, but that's certainly something that, the field is investing a lot in trying to develop these kind of measures.
Yeah, well, going back to the parabiosis experiments, which is just absolutely intriguing, given that so many different organ systems seem to rejuvenate. Have people looked at their epigenetic state with those same tools, or were those studies done before we knew about the epigenetic clock?
So people have gone back to use the epigenetic clock in these kinds of experiments and shown resetting to some degree of the epigenetic age. So the epigenetic clock does track in some of these cases with the apparent age, measured physiologically or some other way. And as Matteo was saying, that's really kind of getting at this issue of, is there a biological age that we can measure? Is it being reset? And that the epigenetic clock has tracked this in these kind of rejuvenation experiments.
Right, right. And then turning to Jing, so you mentioned mentioned rapamycin. Actually, both of you mentioned rapamycin, and it makes sense that people looked at rapamycin, given that it targets the enzyme that is so critical for detecting energy and controlling energy production. But how about alpha K? How did you come up with that?
That's a very good question. Actually, it was partially computationally because we know that starvation and calorie restriction could extend lifespan, and we wanted to get metabolites that could do that. But when we first started, I don't think there's any metabolomic data on the long-lived species or mutations. And actually, in hindsight, if there were, probably we're not going to find alpha KG that way because it's very difficult to assay, even up till today. But what I looked at was essentially publicly available data from gene expression profiling, looking at what happens when you treat cells with starvation or when you inhibit TOR, these kind of things. And one thing that popped out was that they highly regulate the enzymes in the TCA cycle.
Hmm.
Probably, to compensate for the lack of nutrients. But at the time, it looked really promising that perhaps by turning up the TCA cycle, there is a certain metabolite in the TCA cycle that could be responsible for this. And because alpha KG is the rate limiting, component there, so that's where we started to look at.
Mm-hmm. Interesting. Yeah, so now we have two very potent molecules that you've described, but there are so many biological effects in these parabiosis studies, Tom. So do you think that they're mediated by those molecules? No, there must be something else going on. What is it? Cells? Is it molecules? What are the mice sharing?
I mean, our assumption from the beginning, and there's data now supporting this, that there are just factors in the blood that change with age. And in a sense, so and these are, we think these are proteins. So there may be metabolites like what Jing studies, and we study this as well, but we think there are proteins that are promoting youthfulness and resilience in young animals. And then with age, we know we accumulate proteins in the blood, things that are called like cytokines that are signs of inflammation. And they definitely have negative effects on cells. So we think that there really is this crosstalk between, in these parabiotic experiments, young and old animals in terms of what's in the blood. And this has been replicated by using just blood transfusions in animals as well. So serial transfusions of young blood into old animals or old blood into young animals gives similar kinds of rejuvenating or pro-aging effects. So we're all interested in trying to find out what's in the blood that has the, can we neutralize what's in the old, or can we promote or add what's in the young? Now, I'll just add one more quick thing is that, we do think that these proteins that are having these effects are being mediated through the same kind of pathways that are affected by rapamycin or alpha-ketoglutarate, that they're coming to some kind of final common pathways of cell metabolism that are really leading to the changes in cellular age.
Right. So interesting. Yeah. So Matteo, you mentioned that there have to be these high-dimensional, very complex assays, but I imagine in doing these epigenetic studies, maybe there's some locations in the DNA that are actually more informative about aging than others. Do you really have to look at the whole genome, or can you just simplify this test?
Well, what we think is that the regions in the genome that are most associated with aging are actually the least functional. So in a sense, you can think of them as regions that are silenced. They have no effect on transcription. And so changing those has no functional phenotype on the cells. And so the cells have no incentive to maintain these regions. And so what we're seeing is some kind of drift that's occurring in regions of the genome that are not harmful to the cells. And so in that sense, these are great readouts because they tell us about the overall state, how much drift has occurred and potentially how rapidly this drift is occurring. And that's clearly associated with lifespan. But reversing those may have absolutely no effect on lifespan or metabolism. So, in biology, and in life in general, correlation versus causation, right? So we're finding things correlated with lifespan, but certainly not things that we have shown or others have shown can cause or either lengthen or shorten lifespan. As you heard today, the things that lengthen and shorten lifespan that we know of are really have a core impact on metabolism or some aspects of cellular function, autophagy, mitochondria. Those are things that clearly can slow things down. And so that's, I think, where most of the research is. Although one caveat which we haven't discussed is there's also research to reverse aging using transcription factors that can reprogram cells from sort of, older states to younger, more stem-like states. So that's also a big area in the field that we haven't touched on today, but a huge investment is going on in that space as well.
Right. Right. Oh, here I just saw an interesting question here. So given that you have these assays for the epigenome and DNA, people have been identifying DNA from ancient humanoids, including Neanderthals and so forth. Can you use this assay to actually figure out when those people died, how old they were?
Yeah, there are certainly attempts to use, DNA methylation for forensic purposes to figure out the age of the individual. I have not seen it applied yet to ancient DNA, although in principle it could. There may be some technical limitations there, but I think the answer is probably yes.
Yeah, interesting. So another question that popped up is metformin, another drug that people have been excited about. Any comments on that from any of you?
Well, I know quite a bit about this drug.
Great.
So metformin is, of course, a drug that is taken by millions of people for diabetes, and there's some very compelling epidemiologic data that that metformin might extend lifespan in humans. And again, it's not cause and effect, but it's correlated. And the data, basically, it's from Scandinavia, said that people who have diabetes and taking metformin could actually live longer than people who don't have diabetes. So, so that's not a study of healthy people taking metformin, but it suggests that metformin has, in addition to treating diabetes, has an additional benefit in terms of health span, lifespan. And it's not as potent as rapamycin, but there are studies in experimental animals of increases in lifespan with metformin. So just like rapamycin, but actually far more than rapamycin, people are taking metformin who don't have diabetes because they believe that it will give them this benefit in terms of lifespan extension. We just don't have any control trials in humans to show that's true.
Although they are starting the NIH is starting a massive trial to test metformin over the next few years. So, and that's the big problem, is how do you test aging drugs in humans? It's an expensive and difficult proposition, but I think some of these newer trials with metformin may start to get at those answers, right? It'll take
it does get to what Matteo was saying, though, that those trials don't have a good readout of aging, so that those trials are actually using onset of diseases, and that might be useful, but it won't be exactly the same thing, because you just can't do clinical trials in humans for their lifespan. There's just no way to fund a trial like that. So it'll be tough to get the data, but we need some real clinical trials to know whether these drugs are beneficial to otherwise healthy people.
And one last point I want to make is back to other species, there are now interesting companies doing these trials on dogs. So giving, dogs things like rapamycin or similar types of. So apart from mice, which, are relatively easy to, extend and adjust their lifespan, there is a lot of interest in other kind of these intermediate species. And so we'll see if some of these, we may get answers on things like dogs faster than we will with humans, for sure.
Well, I was related to this. I was struck by Jing's work on, using C. elegans, this little worm that has a lifespan of 2 or 3 weeks, maybe, or 4 weeks. I can't remember. And so you can do those studies so much more rapidly. And some of these things are conserved, right, Jing?
Yeah, so the main reason we use it, of course, it's because it's fast to do those lifespans, as Alex mentioned. Also, some of the very fundamental processes are all very conserved, from worms to human, like the mitochondrial components, the mTOR pathway, AMPK, autophagy, everything is conserved. So we like to use it as a sort of a whole system, whole animal test tube, if you will, to look at if anything would have a beneficial effect on lifespan. And then, we will then go back to mammalian cells to use darts to identify the targets, because what we're really interested in is something that's conserved between across species that would be useful for human.
Yeah, so interesting. Well, I think this is really a very exciting area of research where so many different approaches really are making rapid progress here. And I'm really glad that we were able to host this webinar to sort of actually get on, help us communicate to the public, but also with each other about these different approaches. So I'm sure there will be more progress in the next few years that we'll be able to share then. So with that, I think our time is up, our hour is up, and I'd like to thank you, our panelists, for your presentations and our discussion. And I'd like to thank you, the audience, for joining us and for this presentation on aging. And I think you've seen that it's a really wonderful example of collaborative research that is happening in different departments across the campus at UCLA. And I'm really pleased that the Institute for Quantitative and Computational Biosciences, primarily through Matteo, who is a key member and leader in this field, has brought such interesting researchers together for today. So the webinar, of course, was recorded, and there will be a post-event email that is sent to everyone that allows you to watch the presentation, and you're free to share that with others. And with that, I'd like to thank you again for joining us tonight, and have a good evening. Bye-bye.
Bye-bye. Thank you.